we are on the main part of this forum. The head of the Nutritional Genomics Unit will share to us answers to what we perceive as commonly asked questions. Gaya po ng alam natin, ang FAQs o Frequently Asked Questions ay ang go-to natin sa mga websites when we crave for quick answers to top-of-mind questions. Our speaker this morning plotted her presentation in a way that she will cover the basic queries we have about nutritional genomics. Pinakauna dyan, siyempre, ang tanong, ano ba ang nutritional genomics? Our presenter, Ms. Marieta P. Rodriguez, is the Senior Science Research Specialist of the Nutritional Genomics Unit at DOST-FNRI. Ms. Rodriguez, a proud Tomasian, has a degree in medical technology and was able to finish her coursework and Master's of Science in Microbiology. Both were taken from the Pontifical and Royal University of Santo Tomas. Her dedication to nutrition research spans almost 4 decades, with her earlier works focusing on studies on energy and nutrients and nutrition biochemistry. She has been the Senior Science Research Specialist of the Nutritional Genomics Research Unit since the inception of the research unit in 2011. As the head of Nutritional Genomics Research Unit, her current work has centered around the genomic profiling of nutrition and NCD-related polymorphisms, alongside her supervision of both the R&D and the laboratory operations of the Nugen Lab. With a decade-spanning research career, our presenter has authored and co-authored several peer-reviewed articles. She is currently overseeing the implementation of 6 research and development projects of the Nutritional Genomics Unit, with areas ranging from population-based nutritional genomics to translational nutrigenomics and metabolomics. Ladies and gentlemen, I'm now giving you our speaker this morning to speak about anong ganap, pagpapakilala at mga kasalukuyang pananaliksik sa nutritional genomics, Ms. Marieta P. Rodriguez. Over to you, Ma'am Marieta.
Thank you, Jacobs, for that kind introduction. Sa atin pong mahal na director, Imelda Angeles Agdepa, kay Dr. Milflor Gonzalez at sa kanyang team, at sa ating mga mahal na participants. Isang magandang umaga po sa ating lahat. Ngayon pa lang po ay nagpapasalamat na kami for showing your interest to attend this webinar. Let me share my screen. Ang topic ko po ngayong umaga ay tungkol sa kung anong ganap, pagpapakilala at mga kasalukuyang pananaliksik sa nutritional genomics. Hindi nakashare. Hindi ko siya maano. Wow.
Mayet, ipindot mo yung screen share, tapos sa slideshow.
Ito na lang.
So, yung screen share, yung may arrow, and then pindot mo yung slideshow.
Okay, sandali lang po. Okay, sandali lang po. We're having technical problems. So, Jacobs will share the screen. Okay. So muli po, ang topic ko ngayong umaga ay tungkol sa kung anong ganap, pagpapakilala at mga kasalukuyang pananaliksik sa nutritional genomics. Next. Next screen. Next slide, please. So before I start with my presentation, allow me to present the outline of my presentation that I'd like to run through with you in the discussion of nutrigenomics. I will be giving a short background on genome, DNA, and genes, approaches in studying gene and disease, from genomic insight to nutritional applications, and then we will be introducing our Nugen lab and the future prospects. Let me give you a short background of the DNA, which is the molecule of life. Our body is made up of trillion of cells, and these cells in our bodies contain 46 chromosomes packed tightly in the nucleus. We inherit one of its chromosomes from our biological mother and the other one from our biological father. The chromosomes are composed of long, tightly coiled molecule, which is called the DNA or the deoxyribonucleic acid. Ito ang mga materyales na matatagpuan sa loob ng ating katawan. The DNA is made up of chemical building blocks abbreviated as the A, the C, T, and G. The entire length of the DNA consists of these 4 blocks in different combinations, at ito ang mga bumubuo ng mga genes. Ang gene ay isang segment of the DNA molecule that contains the instructions for how, when, where your body makes each of the many thousands of proteins required for life. The way genes are spelled makes all the difference, and one letter out of place in a gene can cause a disease. Next slide, please. So now, since we said food is one of those that turn on and off genes, given persons' polymorphisms, I give you this question. Wouldn't it be wonderful if we could prevent diseases from developing just by changing our diets? Next slide, please. Alam natin na ang mga genes ay pwedeng ma-turn on and off. And proteins are the building blocks of life for our body to develop and to function well. The proteins are also the key on how the body performs its functions properly, and with the food, water, and exercise affecting these genes, ito ang makakapag-turn on and off ng ating genes. Next slide, please. Ano ang polymorphisms? Poly means many and more forms. So one of the most component we look in nutrigenomics are polymorphisms. Gene polymorphisms are the slide chains. Dito pabalik yung slide. Gene polymorphisms are the slide chains in genetic code that are present in at least 1% of the population. These polymorphisms lead to different processes in the body. When the change affects only one genetic letter, this is what we call the single nucleotide form polymorphisms. Next slide, please. So, as you see in this picture, This is a library. The words and sentences, when you put together, can create books that will fit the entire library. Books contain information and we have to read it, right? To maximize the use of these books, especially if we want to ace an exam. Similar to a book, the DNA has all the information the human being has. And to really understand it, we need to carefully analyze it. Now, technology has made it really easy for us. Next slide. The technology enables advances in nutritional genomics studies, and this focuses on targeted next generation sequencing, or what we call the NGS. Next slide. The modern strategies to determine the level of influences or relationship of genes to a certain disease, like the NCDs, are named. And these are the candidate gene studies and the GWAS. Both paradigms have been successfully employed to identify genotype associations with disease. Of course, it made possible with highly advanced genomic technologies. Next slide. So as we see in this picture, the intent is to look at the genes as possible culprits of non-communicable diseases. Next slide. We start with the candidate gene studies. This approach focuses on associations between genetic variations within pre-specified genes of interest and the disease states. Next slide. In contrast to candidate gene, the GWAS scans the entire genome for common genetic variation in the hope of finding genetic markers associated with the disease and may or may not have direct biochemical pathway relationship with the disease state. Next slide. So moving on, the genomic medicine into routine clinical care, it has become smooth because of the two genetic association tests mentioned earlier. Next slide. So, next slide, please. With the realm of nutrition and preventive medicine, we have nutritional genomics, or the application or high throughput genomic tools in nutrition research. Next slide. In studying genomics, we will be able to identify genomic markers, stratification of patients for clinical trials, or we can determine the resistance of a certain markers, or we will be able to establish combination therapy, and it can assist us in resurrecting failed trials. Next slide. So we are now entering a new era in the field of food and nutrition research, and this is called the nutrigenomics. It is believed that nutrigenomics will revolutionize wellness and disease management. Next slide. With the realm of nutrition and preventive medicine, we have nutritional genomics, or the application of high-throughput genomic tools in nutrition research. Nutritional genomics is defined as the interface between the nutritional environment and cellular or genetic processes. It presents how food affects metabolic pathways and homeostasis, and how genetic differences affect individual response to nutrients. Because of NGS and high-throughput sequencers, it is now possible to analyze certain gene changes in a global and or system-wide perspective. Next slide, please. Do you know that the main goal of nutrigenomics is to identify genes that can be targeted by nutritional intervention to prevent diseases from developing? Next slide. Next slide, please. But we cannot choose our family or genetic risk that they pass on to you, but we can do something about it if we choose our diet. Next slide, please. Between nutrigenomics and personalized diet, everyone responds to different food that we take, and a diet may be optimal for one person, but may predispose another to a certain type of diseases. And individual requirements for different nutrients can vary based on a person's genetic makeup. So nutritional genomics is defined as the functional interaction of diet and food with genome at the molecular, cellular, and systemic level, which may lead to personalized nutrition in other technical term, or other technical term is precision nutrition. Both have the same ultimate goal to design tailored nutritional recommendations to treat or prevent nutrition or metabolic disorders. Nutrigenomics describes how our inter-individual differences in DNA composition actually creates variation in our protein function. Some of them are enzymes involved in metabolism. This further explains, as opposed to another who seldom, certain individuals, even though they eat a lot and don't get fat easily, as opposed to another who seldom eat but gets fat easily. The other aspect of nutrigenomics is that it describes how nutrients broken down from various food can help maintain integrity of our DNA to perform its normal function. It is eating based on our personal genes, hence more specific and targeted. Next slide, please. So knowing our genes can guide us in choosing the right food, lifestyle, and goals. How then our genes impact our well-being at the personal level? Between two people, there are only 0.5% difference in DNA. And this small 0.5% up to 3 million letter changes is what makes us unique. And only one letter difference is enough to make the big change. As you can see in this slide, when the I changes to A, you can see here the consequences. Next slide, please. So the triple burden of malnutrition, which involves overnutrition, undernutrition, and micronutrient deficiencies, increase the various health problems in our country. In the Philippines, results of the 2013 NNS in the 2018 expanded ENNS revealed vitamin A deficiency is about 20.4% among 6 months to 5-year-old. Anemia is 9.7 among female of reproductive age. And children under 5 years of age, 30.3% is stunted and 5.6 is weighted. For the adult population, 19.2% is considered as hypertensive, and the prevalence of type 2 diabetes mellitus is about 8.2%. And 4 out of 10 adults are overweight and obese. Next slide. So the Food and Nutrition, being the lead agency in conducting surveys, capitalizes on the R&D program on nutritional genomics as a solution to triple burden of malnutrition in the country. Next slide, please. Advances or advancement in food and genetic technologies exemplify scientific paradigms in the field of nutrition. The formulation and commercial development of functional foods provide enormous health benefits by reducing the risk and enhancing the ability to manage lifestyle and related diseases, thus improving the quality of life. Simultaneously, nutritional genomics substantiate these health claims by elucidating the basis for marked differences in individual responses to dietary factors. Next slide, please. Some of the potential benefits of nutrigenomics are the increased focus on healthy lifestyle and disease, motivate positive behavior changes, and increase awareness of risk of certain conditions. This is more effective in assisting or motivating patients and consumers to make or sustain appropriate healthy changes to their usual diet and lifestyle, which will lead to reduced health care costs, and it may be disease specific, and we can avoid the hit and miss for certain types of diseases. Next slide, please. So now, the supermarket today may be our pharmacy in the future. The convergence of functional food and nutritional genomics enables contemporary kitchens and supermarkets to become pharmacies of tomorrow. This is according to the study of Ferguson. Why go to the drugstore and buy expensive medicines when we can just trim down our carbohydrates and sugar by buying the right kind and amount of food in the supermarket? Testimonies have already been made by patients who were able to discontinue their intake of medicine after eating foods of low glycemic index or low sugar. So the Food and Nutrition Research Institute conducted researches on nutritional genomics under their R&D program. Next slide, please. Since its inception in 2010, the nutritional genomics R&D has found home in the premises of the DOST-FNRI. Next slide. Next slide, please. We also take pride in establishing the Nutrition Laboratory as it continues to grow in scope. Next slide. And we will continue to explore our horizons and grow quality service to nation building. Next slide. So how we do genotyping? The analysis of complex organisms from different sources, such as the whole blood, through the application of molecular biology techniques, requires the purification of pure, high molecular weight genomic DNA. Different technologies and methods are available for isolation of genomic DNA. In general, all methods involve disruption and lysis of starting material, followed by removal of proteins and other contaminants, and subsequent recovery of the DNA through elution. The concentration and purity of our eluted DNA is assessed by spectrophometric analysis, while DNA quality is assessed by visualized through agarose gel analysis. The eluted DNA is ready for use in most downstream applications, such as genotyping of single nucleotide polymorphisms by high-resolution melting melt. High-resolution melting melt is a simple, rapid, and low-cost mutation scanning method. Its advantage is the fact that PCR amplification and melting curve analysis are performed within the same tubes or plate. This feature is particularly important for a routine diagnostic setting. HRM is based on computer analysis of DNA melting transitions, whereby it is possible to record more than 25 readings per 1 degree centigrade by monitoring the change in fluorescence that results from gradual temperature-dependent release of a saturating DNA binding dye. Next slide. Here, we are now in the process of positioning our nutrigenomics laboratory quality management towards ISO IEC 17025-2017 standards. This will allow the laboratory to demonstrate its competency in every aspect of its management system and technical operations. The presence of quality assurance through the use of appropriate operational procedures guarantees laboratory personnel, top management, and customers that errors are minimized and corrective actions are taken when non-conformities are identified. Next slide, please. In its journey towards becoming a competent molecular biology testing laboratory, we can empower the Nutrition Lab as an in-depth service provider of molecular biology techniques and develop more protocols that can sustain and expand the scope of accreditation. Next slide. Our laboratory is composed of competent personnel with various academic backgrounds on molecular biology, and biotechnology, genetic counseling, biochemistry, medical technology, and microbiology. We see to it that competency requirements are identified prior to release of authorization for the technical staff of the Nutrition Lab. The competency assessments are done periodically. Next slide, please. Here, we see to it that all the requirements of a laboratory or the technical competency of the Nutrition Lab are in place. Next slide, please. In doing our R&D researches, we are in partner and evaluated the assurance by comparing it to the results generated from various accredited laboratories such as the Microgen or the UMPI Medical Molecular Institute of Malaysia. Next slide, please. The quality assurance is evaluated by comparison of results generated from a single sample versus the two ISO IEC 17025. And this method being validated is being tested for its precision, accuracy, robustness on analysis, annealing temperature, primer concentration, and template concentration. Next slide, please. Among our partners in the conduct of our R&D researches includes the National Institutes of Health, the National University of Singapore, the Philippine Genome Center, the NUGO, and APNO, and the UMBI. Next slide, please. So some of the projects conducted by the FNRI in our Nutrition lab were the following. Next slide. We have conducted evaluation of the association of FTORS 9939609 to body mass index, physical inactivity, and dietary intake. Here, the results try to explain why certain individuals, even though they do not eat a lot or even less the recommended intake, still get obese, especially if they have or if they carry the A allele, which is the risk allele carrier. Next slide, please. Another study is on the polymorphisms of FTO and GHSR genes, which affect subjective feelings of appetite among Filipino adults. Another study, next slide please, is the difference in mean fat intake based on CD36171667 genotypes among hypertensive and non-hypertensive adults. So here in this study, results showed that hypertensive men in the age group of 20 to 39 years old who are carriers of A allele have higher BMI and fat intake. Next slide, please. In the study on the differences in mean sodium intake based on ADD1 RS 4961 and TRPV1 RS 8065080 genotypes among hypertensive and non-hypertensive adults, the results showed that the mean sodium intake among hypertensive women belonging to the 20-39 years old who are carriers of the G allele of ADD1 RS4961 are higher compared to those who have the T allele. The mean sodium intake among hypertensive men who are carriers of T allele are higher compared to those who have the C allele. Next slide, please. In the study on eating behavior and known obesity associated genes in selected 9 to 12 years old children in Taguig in Pateros, the results showed that obese children carrying the risk allele of FTORS 9939609 and RS 1421085 have higher food responsiveness and lower satiety responsiveness. In our study, next slide please. In our study on malunggay leaves powder as tumor suppressor for colorectal polyps, done on spray dolly rats, this showed that the diet consisting of malunggay leaves powder consistently reduced the mutation frequency of KRAS oncogene in rats after 11 months of intervention. So this also, the study also showed that the malunggay leaves powder helped in the progression of the colorectal polyps. Next slide, please. In the study on effect of brown rice on the biochemical indication of blood glucose levels and the expression of the TCFL2, the GLUT4, and the SLC2A4 genes, which is conducted among government employees here in Taguig City. The study shows that consumption of brown rice for 6 months generally improve all metabolic parameters that we investigated except for the lipid profile. It also shows that male government employees carrying the CC genotype who consumed brown rice for 6 months had increased expression of TCF7L2, and it was observed to have lower blood glucose levels. So moving forward, the DOST-FNRI continue to capitalize on R&D projects and continue to invest on the best application technology and state-of-the-art equipments. In this slide, we can see that we can empower the NuGen Lab as in-depth service provider of molecular biology techniques. We still need to strengthen the capabilities of the laboratory personnel in terms of providing more services to our internal customers or potential external customers. In this way, we can develop more protocols that will sustain and expand the scope of testing and scope of our accreditation. By the way, we are now in the process of acquiring our accreditation. Our application has been submitted to PUB, and we are just waiting for them for the surveillance. Next slide, please. So the main function of the diet is to provide the necessary nutrients to meet our nutritional needs. There are evidence that the relationship between diet and health arose from the concept of functional foods. In general, functional foods are those that are not only fulfill nutritional function, but also contain biologically active components that offer high benefits and reduce the risk of suffering diseases. Knowledge of nutritional genomics can revolutionize how functional foods are developed. And this technological breakthrough could eventually make it feasible to tailor a diet for an individual's specific genetic profile. Nutrigenomics will have a profound effect on future disease prevention efforts, including the future of the functional food industry. Next slide, please. So with the growing field of artificial intelligence or the AI, nutrigenomics is becoming more popular and customer-facing. Specialized DNA tests focus on various aspects of a person's genetic information, microbiome, lifestyle, and environment, yielding to a personalized diet plan created from billions of data pieces about the customer. This test provides nutritional guidance based on the nutrigenomic whole system approach, a big improvement from generalized diet recommendation. Analyzing complexity of personal health metrics has become possible due to advances in artificial intelligence technology. In DOST-FNRI, researches are being developed and optimized applying AI in order to not only predict, but may also lead to provide personalized diet recommendations for specific diseases such as hypertension and obesity. Wait lang. Next slide, please. So another project that the FNRI doing is the establishment of a personalized genotype-based nutrition and life. Sorry for that. In the implementation of this project, the convergence between the quality of procedures set by the technical working group and the panel of SNPs identified during the first and second phase of this program will result into the genotype-based nutrition and lifestyle recommendations, which will be subjected to an RCT. From an RCT, a set of recommendations will be derived, and this will be labeled as the My Gene, My Diet. This is a comprehensive genetic test consisting of a panel of genetic markers of a specific health outcome. A personalized nutrition and lifestyle recommendation will be rendered when a biological sample of an individual is tested using this panel. Next slide, please. So in genomics in healthcare, the development of new diet and diet recommendation and a new way of looking at diseases once we establish the role of one's genetic makeup, the role of environment, lifestyle and dietary habits, past experiences, and epigenetics and microbiota. We must remember that it's not nature versus nurture. It is nature and nurture. Next slide, please. Okay, thank you for your attention.
All right. Salamat po, Ma'am Mayet. Thank you. Stop share, no? Okay. And so with that, maraming salamat po, Ma'am Mayet, for that presentation. May you hold your horses at this point. Synthesize muna natin, and we'll be opening up the open forum later on. So before natin i-play yung video for the ABP, kasi kanina tinutukoy ni Ma'am Mayet yung Nutrigenomics Lab or the NuGen Lab, no? So we'll be sharing a short clip about that laboratory later. But allow me to synthesize yung naging discussion ni Ma'am Mayet. So as I see it, as the moderator, nakikita ko the presentation of Ma'am Mayet is actually divided into 6 segments. No? The part 1, she discussed about the central dogma. Basically, the research on nutritional genomics here at FNRI were actually focused on the genomics part, on the DNA part. No? We're looking into the polymorphisms. So nag-discuss siya ng mga polymorphisms wherein she said a minor change or a change in the nucleotides could actually lead into, sabihin na natin, changes or alteration on how the protein will be translated later on or kung paano na yung mga phenotypes. The focus of the studies that were conducted at FNRI, yun na po yung part 2, is actually as a response to the increasing micronutrient deficiencies and the NCDs. Kaya pumunta po siya doon sa triple burden of malnutrition. So basically, the nutrigenomics research of FNRI is actually capitalizing on the triple burden of malnutrition. On the part 3, she actually discussed the current technologies na pwede nating gamitin to elucidate the polymorphisms. Meron siyang sinabing next generation sequencing, and she also talked about targeted and untargeted GWAS, current endeavors, including the establishment of the Nugen lab. So sa part 4, she actually discussed about the capacities, the capabilities of the Nugen lab. The Nugen lab is actually housed here at the DOST-FNRI at this point in support of the research and development initiatives. Kaya narinig niyo po kanina, is actually talking about internal clients. But of course, as we expand and we actually welcome, no, as part of this next video that you'll be seeing later on, to open the laboratory for our, sa atin po, na mga gustong mag-research about nutritional genomics. On the part 5 of her presentation are actually the previous studies that were conducted through the 10-year span of nutritional genomics research here at FNRI. Kung napapansin niyo po, we're actually focusing on obesity, nutrigenetic studies on eating behavior, sodium sensitivity, salt sensitivity. And at the same time, there are attempts on what we call the nutrigenomics part, yung hardcore. Meron tayong gene expression analysis sa mga diabetics. And at the same time, meron po tayong ginawa study on Moringa oleifera, which is being tested on animal model. And on the last part, meron po siyang mga tinukoy na mga way forward o yung mga gustong gawin ng nutritional genomics in the future. Meron tayong study that was completed on artificial intelligence. On artificial intelligence, meron tayong hypertension risk predictor and a current endeavor on what we call the DNA-based nutrition and lifestyle recommendation, which is currently ay sasagawa po natin randomized controlled trial wherein we are going to compare a standard nutrition advice and a DNA-based nutrition advice. So basically yun po yung synthesis ng naging presentation po ni Ma'am Mayet Rodriguez. So before we proceed with the video, let me just acknowledge po yung mga kasama po natin ngayon dito sa Zoom. We have our former Chief of the Nutrition Research and Development Group, Dr. Celeste Tanchoco, our current Chief, Engineer Rosemary Garcia, our former Deputy Director, Dr. Aida Aguinaldo, and our current Deputy Director, Dr. Anthony Calibo. Maraming salamat po sa pagsama niyo po sa amin ngayong umaga. And now, allow me to share to you a short video, an audio-visual presentation of the Nugen Laboratory. Bago po tayo mag-proceed sa question and answer portion.
In 2010, DOST-FNRI revolutionized its take on nutrition by incorporating genomics in its researches. Investigating the relationship of anthropometric, biochemical, clinical, dietary, and health components to the genetic makeup of Filipinos, DOST-FNRI is looking into providing a more personalized approach of achieving optimum nutrition to every Filipino. Through nutritional genomics, we attempt to better understand how a single nucleotide, a single letter, can make a big difference in our way of living. And so, to support this endeavor, DOST-FNRI capitalized on establishing its molecular laboratory known as the Nutritional Genomics or the NuGen Lab. With its thrust of bringing science for the people, the NuGen Lab provides assessment of genes and genetic variants associated with micronutrient deficiencies and diet-related non-communicable diseases. The NuGen Lab is equipped with some of the state-of-the-art equipment in molecular biology, such as the automated nucleic acid extraction machine, bioinformatics, real-time and digital PCR systems, automated liquid handling system, micro-volume spectrophotometer system, and gel documentation system. With a complete workflow adhering to the highest standard of competency in laboratory testing, NuGen Lab is setting the mark in providing mole biotechniques with transparency, validity, and reliability. Whether you are an individual or a group of enthusiasts seeking answers in nutritional genomics, the NuGen Lab awaits you. Our proficient personnel works in close contact with our customers through customized analysis, provision of technical consultancy in nutritional genomics, and standardization or laboratory competency. To even serve you better, we are partnering with a leader in genomic testing for a higher throughput and faster turnaround time. Welcome to the new generation of nutrition. Welcome to the NuGen Lab.
Alright, there you go. Yun po ang pasilip sa ating NuGen Lab, ang Nutritional Genomics Laboratory ng DOST-FNRI. As a support, doon po sa sinabi ng ating director kanina, we are welcoming collaborations pagdating po sa research and development initiatives on nutritional genomics. At this point, dumako na po tayo sa ating open forum. Babasahin ko lang po yung mga questions na nandito ngayon sa ating chat box. Alright. The first question is from Sir Lyndon Plantilla. His question is, can middle-aged people can still reverse diabetes and diabetic and hypertensive conditions can be changed by changing diets? How does NuGen or FNRI proposes to do it? Yes, Mamayet. You may open for your audio. So the question po is, can middle-aged people can still reverse diabetes and hypertension, or diabetic and hypertensive conditions can be changed by changing diets? How does NuGen or FNRI proposes to do it?
Maraming salamat sa iyo, Lyndon Plantilla, sa iyong katanungan. Para po sa akin, ang pagiging diabetic and hypertensive ng isang middle-aged na tao ay pwede pang, the progression, we can stop the progression of this disease into a more serious condition, but by eating the right food and the right amount of diet. Pwedeng mananatili pa rin sa isang individual ang pagiging diabetic dahil siya ay nasa middle age na, no, mahihirapan tayo na ma-revert ito or mahihirapan tayo. It will take time before we can lower yung ating condition ng isang diabetic. Sa wastong pagkain and living a healthy lifestyle by doing some exercises, we can stop the progression of this disease into a more serious condition. Posibleng it will take time na mabalik tayo sa normal. But as long as we are eating the right food and we are following the right diet, living a healthy lifestyle, masasabi ko po na pwede nating ma-normalize ang ating diabetes. Depende pa rin po ito sa genetic makeup ng certain individual.
Sige. I hope you Mamayat was able to answer your question, Sir Lyndon. Actually, coming from the perspective of genomics, ang ginagawa nating research on nutrigenomics just is more on the preventive side of the continuum of healthcare, no?
Yes.
Of course, we're looking into the polymorphisms, titignan natin yung risk. But of course, kung nandyan na talaga yung condition, gaya ng sabi ni Mamayat, it can somehow prevention pa rin yung importante. Okay, sir? The next question is from, okay, let me just entertain one question po sa Facebook. We have a question from Sir Christian Flores para po ma-include naman natin yung mga kasama natin na nasa social media platform ng FNRI. Ayon po kay Sir Christian, is the Nugen Lab already offering genomic services for public consumption? Mamayat. There's a question from Sir Christian Flores, and that was given to us via the Facebook Live.
Thank you, Christian. Sa ngayon, ang Nutrigenomics Laboratory ay hindi pa siya open sa public. But we have plans of opening it up para ma-accommodate namin yung mga nagnanais na magpagawa ng genotyping dito sa amin. Right now, we are still working on the accreditation ng aming laboratory to ISO 17025 2017 standard. At pinipilit namin or ginagawa namin ang aming makakaya para ma-open na ito sa public sa pinakamurang halaga. You know, doing genomic studies is very expensive. And gusto namin na kung i-offer namin ito sa publiko, naka-establish na sa amin yung mga tamang proseso ng aming mga ginagawang technologies. And then, kung saan pwede kaming makatulong sa public by charging minimal fees. Pero sa ngayon kasi, hinahanap pa namin yung paraan kung paano namin mapapababa ang mga charges na pwede naming hingin sa aming customers. But let's remember talaga na doing genomic studies is really very expensive. On our part, yun din ang aming nagiging problema dahil hindi naman malaki ang pondo na nakukuha natin. Pinaghahati-hati lang namin in the conduct of our R&D researches. But soon, we will open our services to the public.
Right? Para maibigay natin yung to answer your question, sabi nga ni Mamayat, bubuksan siya soon sa public. And then looking forward po na may offer yung mga test na to actually the Nugen lab is actually building an expertise on genotyping single nucleotide polymorphism. So at this point, nandun pa po siya sa stage na ay naayos natin yung system. But looking forward po na ito ay ma-offer na po natin sa ating mga kababayan. Balik po tayo sa mga questions dito sa Zoom. This is from Ma'am Sigundina Devota Dilao. I'm wondering how much it will cost. Ano po kaya yung sinasabi ni Ma'am Sigundina na cost? Is it the cost of the testing? Well, basically, sige, mamayat, cost daw po. Maybe it's on the analysis.
Sa ngayon po, wala pa po kaming costing ng analysis na pwede namin i-offer sa public. Pero based po dun sa mga ginagawa naming projects, dahil po sa mahal ng mga reagents and mga consumables na ginagamit sa pag-a-analyze ng ating DNA, should I say this? Kasi based on our computation, per analysis of our sample, sa ngayon po dun sa mga researches na ginagawa namin, Jacobs, should I say more than 25,000K yung analysis?
Figure na po.
Figure na. Pero yun po ay sa mga researches na ginagawa namin. Kaya po ginagawan po namin ng paraan para po mapababa. We know naman po na yung doing DNA analysis is not an ordinary analysis. Talaga pong it involves expensive use of reagents.
So yun po ma'am, no? May nagbigay na si Mamayat ng ballpark figure, but looking forward to lower that cost for the analysis. I hope our speaker was able to answer your question po. This is a two-part question from Ged. Tama po yung pronunciation ko, no? Ged or Jed from DOST-STII. Ito po yung unang tanong. Open po ba ang Nugen for individual or walk-in clients for diet analysis or recommendation? Actually, Nugen Laboratory. Okay. Yun ma'am, open po ba ang Nugen for individual or walk-in clients for diet analysis or recommendation? Yun yung unang tanong ma'am. Take note po natin. The second question is, does FNRI has a plan to launch a Yossi Kadiri type of campaign to help the public shift their diet pattern or behavior? Okay. Sige ma'am. I think yung second question po can be answered, I guess, from the communications arm of the FNRI. Although, sige mamaya, I did to pick a brain on this. Yung dalawang question po na ating pong kasama mula sa DOST. Okay. The first question is, open po ba ang Nugen for individual or walk-in clients for diet analysis or recommendation? Okay.
Diet analysis. Actually, mayroon kaming mga kasamahang nutritionist, dietitian dito sa aming division, and they do the counseling. If you mean diet analysis, yung meal ba mismo ang gusto mong malaman? Ayan. Yes, ma'am.
Opo. Ang intindi ko po kasi kanina, parang with certain information ng individual, and it can be analyzed, and then you can recommend us, lalo na kung may sakit yung tao, yung ganun pong klase ng analysis and recommendation. Thank you.
Okay. Thank you po, Ma'am Geraldine. Tungkol po dyan, sa kasalukuyan po ay may ginagawa kaming study nga na kung saan pag mayroon ka ng genetic profile or meron ng genetic profile ang isang individual at nalaman mo na na mayroon kang risk allele, magkakaroon po tayo ng specific diet recommendation para dun sa individual na yon. Ito pong study na to ay ongoing sa kasalukuyan, pero ito po ang aming tinatahak. Once na you know your genetic makeup and you possess the risk allele, mayroon kaming mga nutritionist, dietitian, and genetic counselor who will help you evaluate or who will give you some diet recommendations. Yan po, ma'am. Pero yan, ginagawa pa po natin yon sa kasalukuyan, and hopefully soon din matatapos din natin po ito to come up with this genotype-based diet recommendation.
Right, Ma'am Gayad, maidagdag ko lang po, kasama po sa frontline service ng DOST-FNRI ang pagkakandak po ng counseling, nutrition counseling. So at this point, yun po yung na-offer pa ng ating institusyon para sa mga walk-in clients. Meron po tayong nutrition counseling. But of course, given the pandemic that we have now, may mga restrictions po tayo. And yung pong sinasabi ni Ma'am Mayet na dina-develop na DNA-based nutrition recommendation is complementary to the nutrition counseling po na ibibigay or binibigay ng DOST. Okay. I hope I was able to answer your question, ma'am.
Second question.
Moving forward, this is a question from Sir Eric Sagaysay. What is the difference of... Sorry, nakamute pala ako. So ito po yung question from Sir Eric Ruiz Sagaysay. What is the difference of genetically made foods and organic regular foods when it comes to the effect of lifestyle diseases? So, medyo controversial yung tanong ni Sir. It's more on GMOs, the difference between GMOs and organic or regular foods when it comes to the effect of lifestyle diseases. Mamayet, I think Nugen hasn't conducted any
GMO studies.
GMOs.
Yes, I hope I can answer your question. Pero as far as I know, genetically engineered pagkain ay you nag-i-introduce ka ng a certain type of what do you call organism, bacteria or whatever to a certain food para it's either to improve the quality of the product or the food or para din maiwasan na itong pagkain na to ay hindi maatake ng mga pest or virus. Yung organic food naman ay just the natural food na makikita natin sa everywhere without this yung mga organic food products na to ay hindi ginagamitan ng mga fertilizers and they are grown naturally.
Yes, sir. Unfortunately, sir, wala pa pong nagagawang study ngayon ang DOST-FNRI comparing GMOs with the organic foods. But yeah, it's quite an interesting way to move forward. All right, this is another question from our Zoom platform from Marjorie Ann Layosa. I think this is a very insightful question. Mamaya, ito ang tanong ni Marjorie. Looking forward, will individualized diets have any implications on the general guidelines of healthy eating, such as the nutritional guidelines for Filipinos and Pinggang Pinoy?
Ito pong ating individualized genotype-based diet recommendation ay hindi po natin babaguhin yung mga existing nutritional guidelines among Filipinos. Ang karagdagan lang pong idadagdag namin dito sa ating genotype-based ay yung malalaman natin dito ang ating genetic profile. Kasi once na alam na natin ang mayroon na tayong kaalaman sa ating genes, may possibility na maaari nating iwasan yung mga pagkain na hindi natin dapat kainin. Take for example, yung mayroong mga taong matataba na kain sila ng kain and they keep on getting fat. Pero meron naman din mga tao na kahit na ano ang kainin nila at kahit na gaano kadami ang kainin nila, hindi sila tumataba. This is because ito yung genes na present sa kanila. May possibility na sa isang individual, wala sila nung tinatawag nating risk allele for obesity. Kaya kahit anong kainin nila, ay hindi sila tumataba. Meron din naman mga individual na nasa kanila yung risk. Kaya konting kain lang nila, they get fat. So at nagiging lalo silang obese. I hope I explained.
Okay,
Well.
sige Mamayat. Ganito po. Tama po yung sinabi ni Mamayat, no, yung implications nung pag sinasabi natin DNA-based diet recommendation sa existing, no, ng mga nating recommendations, yung nutritional guidelines for Filipinos in Pinggang Pinoy. Actually po, yung DNA-based nutrition recommendation, if I may just the word, personalizing more the personalized recommendation na binibigay. Well, hindi po ako nutritionist dietitian, but based on the things I've learned sa mga RDs po na kasama natin sa FNRI, yung pagbibigay ng nutrition recommendation is already a personalized interaction between the R&D and the client. So, pinapersonalize natin yung recommendation. But what the DNA-based nutrition recommendation is trying to propose is that given the additional information, genetic information, mas lalo pa nating mas ma-personalize yung binibigay po natin ng mga recommendations. Ano po? And nasabi rin kanina ni Ma'am Mayet yung alleles. Actually, may mga nakita akong question dito. Ginugel daw nila kung ano yung allele. So yung allele po, yun yung version ng genetic information na meron sa isang tao. So may sinasabi kanina si Ma'am Mayet ng mga letters na A, G, C, and E. Ano po? So kapag pinagsama mo yun, nagiging genotype siya o yung genetic type ng individual. Pero kapag yun ay hinimay mo at naging mag-isa na lang siya, for example, meron tayong tinatawag na TT genotype or the mixture of both the thymine sa nucleotides or ANA or AA. So kapag pinaghiwalay mo na yun at mag-isa na lang si A, yun yung allele. Yun yung version, no, ng genetic type ng tao. I hope I was able to somehow give enlightenment kung ano po yung allele na sinasabi ni Ma'am Mayet. No? Meron tayong risk allele na sinasabi niya at meron tayong normal allele. Yung risk allele, usually, yun po yung sinasabi nila na nagkukos ng disease conditions. But of course, I would like to give a caveat na hindi po lahat ng mga risk alleles ay nagkukos ng disease. No? May mga risk. Ang naging focus lang po kasi talaga ng mga pag-aaral sa FNRI, yung mga alleles na sa tingin natin ay kailangan nating bigyan ng attention. Okay po? So I hope Ma'am Mayet was able to answer the questions. Ito pa po Ma'am Mayet, merong nagbigay sa atin ng question. Okay, I'm down to the last 3 questions. Good morning. This is from the University of Mindanao. Magandang umaga po sa ating mga kababayan mula sa Mindanao. Good morning. In conducting genomics-related nutrition studies, may we know if the new gen has its own standard protocols in terms of ethical considerations? So our question is actually capitalizing the ILC, legal and societal implications of doing nutritional genomics research. Ito ma'am, in conducting now the studies, are we adhering or we have our own standard protocols in terms of ethical considerations?
Maraming salamat po sa inyong katanungan. Gusto ko pong ipabatid sa inyo na yung mga R&D researches na ginagawa namin dito sa aming institusyon ay dumadaan sa ethics review before ma-implement itong mga studies na to. Actually, the samples that we are using here is a component of the expanded National Nutrition Survey being conducted by FNRI. The expanded nutrition survey has its own ethical clearance. They were approved to conduct with the enumerated components of the survey. And using this data coming from the survey, we also applied for an ethic clearance prior to the implementation of certain projects that we will implement in our laboratory. Hindi po kami pinapayagan na gumamit ng mga data kahit na kahit na stipulated ito sa National Nutrition Survey. Sa amin pong pagpa-implement ng aming mga studies, inia-apply pa rin po namin ito ng ethics clearance.
All right. So to answer that question, yes, we are adhering to ethical considerations, particularly on ethical way of doing research. But I do understand, of course, yung ating pong question siguro is emanated from the idea that the genetic information is too personal for an individual at stigmatizing minsan. No? Tama po yung mga information na kukuha natin, lalo na sa mga genetics. Alam natin na kapag sinabi natin genetics kasi is namamana siya or talagang naka-embed na sa individual. But of course, the ethics committee of FNRI, lalong-lalo na po sa pag-approve ng mga studies natin na gagawin, is actually adhering to the existing guidelines and regulations when it comes to doing ethical research. Shout out lang po ako kay Ms. Shane Pe, who is actually beaming our activity on the Philippine Event News TV. Maraming salamat po, ma'am. Okay, we're down to last 2 questions. Marami tayong mga tanong. Zoom platform. Okay, last 2 questions. Kung gagawin para sa masa ang services ng NuGen, anong pwedeng maibahagi sa mga LGU-based nutrition offices? So ito na po tayo, ma'am, sa pagtatranslate ng Nutrigenomics. Ulitin ko lang po, kung gagawin para sa masa ang services ng NuGen, ano ang pwedeng maibahagi sa mga LGU-based nutrition offices?
Gaya nga po ng ginagawa namin dito sa NuGen, pwede po nating ma-share sa ating mga LGUs ang specific diet recommendation for a certain individual base dun sa mga results ng ginagawa naming pag-aaral. Sa ganito po, maiiwasan natin na mag-progress pa ang mga certain diseases or ang mga non-communicable diseases dahil mabibigyan natin sila ng mga guidelines or dietary recommendations para makaiwas sa mga nasabing sakit. Actually, dito sa aming mga ginagawang researches, we are working in collaboration with the different LGUs kung saan namin isinasagawa ang pangungulekta ng mga datos para gamitin namin at maka-come up kami ng mga recommendations based on the findings from participants namin coming from the LGUs. At natutuwa po ako dahil kung kami lang naman ang gagawa ng ganito, hindi namin or hindi kumpleto ang aming mga resources, wala kaming mga participants. So we are doing our R&D in collaboration with the different LGUs.
So, ayan, at this point po, yung bottom line ng sinabi po ni Ma'am Mayat is, at this point, nandun po tayo sa part that we are actually asking the local government units to help to collaborate with us in this research. And eventually naman po, pagkatapos po nating ma-synthesize lahat ng mga resulta ng mga studies na ginagawa natin, itatranslate po natin ito sa ating mga kababayan. Okay, before we go on to the last question, shout out lang din po kay Ma'am Bel Susara, who is actually covering us also from the platforms of the Eagle Broadcasting Corporation, Net25, and Easy Radio Aguila. Good morning po, Ma'am Bel. Ito po, ma'am, yung last question na entertain natin. Although marami pa po tayong question talaga na dumadating sa ating chat box. This is from Ma'am Shane Pe of the Philippine Event News TV. This is a 1 million peso question, mamaya. How is nutritional genomics being used to improve health? Actually, this is somehow a question that would synthesize what has transpired this morning. Ulitin ko, Mama, how is nutritional genomics being used to improve health?
Okay. So, gaya ng nasabi ko kanina, nutritional genomics studies the effects of food, effects ng food sa ating genes, and kung paano naaapektuhan ang genes ng mga nutrients na ating kinakain. So, sa ating mga pag-aaral, pwede nating i-share sa ating mga kababayan, especially when we know of our mga genetic makeup or genetic profile, if we possess yung mga risk allele na sinasabi natin, pwede nating gabayan or pwede tayong magbigay ng guidelines kung paano makakaiwas sa mga sakit at kung paano natin hindi man natin ma-stop ang progression or hindi man natin ma-stop ang paglabas ng mga iba't ibang sakit, pwede tayong makatulong sa ating mga mamamayan sa pamamagitan ng pagbibigay ng mga guidelines or recommendations doon sa mga pagkain na dapat nating iwasan o dapat nating kainin. Yung the amount and types of foods na ating kakainin will mean a lot para sa mga individual na mayroon ng nag-e-exist na sakit or dun sa mga individual na wala pang sakit, ito ay makakaiwas tayo na magkaroon pa ng onset ng mga diseases.
Right. So I think yes, Sir Bads, you're raising your hands. Opo, Sir Bads.
Yes, Jacobs, if I may interrupt for a while, I would just like to address the second question of Ms. Jed from our dear friend from the OSTSC.
All right, sir. Sige po. Sige po.
Regarding the plan, if FNRI plans to launch a Yossi Kadiri type of campaign to help the public shift their diet patterns or behavior, we are actually doing this continuously. And yes, you are correct. The negative or type or threat approach to messaging, especially in the media, is most often being noticed by our audiences. And this has been also pointed out by Undersecretary Guevara in our series of campaigns that we have been doing since the pandemic. This FNN Talks on Nutritional Genomics is just the start. And subsequent activities, especially in engaging the media because they are our partners in multiplying our messages on the benefits of nutritional genomics, will follow. And this has also to be balanced out with the positive approach of sending messages because admittedly some people also appreciate the positive messages, and I appreciate your proposal on doing campaign. Do not worry, you will be engaged on this as we move forward with the campaign on nutritional genomics. Thank you, Jet, for your question. Back to you, Jacob.
All right, sir. Sir, maraming salamat po. Tama, yun po yata yung na-miss ko na question kanina ni Ma'am from DOST-STII. Gaya po na nakikita nyo, DOST-FNRI is not only partnering in research, but also we are also partnering and communicating our research. Alright, ang dami pa po nating tanong na nare-receive, but of course, Ma'am Mayet will be happy to answer those questions offline. Ano, Ma'am Mayet? I
yes,
think our,
you can always email us for
yes.
For your queries.
Okay po. So muli maraming salamat po Luzon, Visayas, Mindanao sa pagkapit po sa amin dito sa isinagawa po nating unang attempt para po maipaliwanag sa mas nakakarami kung ano ang nutritional genomics. Kanina binabantayan ko po yung ating mga kasama sa Facebook. Nag-peak po tayo sa 79. Maraming salamat po, and we still have 155 here inside the Zoom platform. Bago ko po ibigay ang hapag sa ating Deputy Director, Dr. Anthony Calibo, para po sa kanyang closing remarks, ating ginawang FNN Talks, OMICSena, mga frequently asked questions sa nutritional genomics, Gusto ko lamang po i-synthesize sa isang mabilisan kung ano po ba ang nutritional genomics. Ang nutritional genomics ay ang kaugnayan ng genes sa nutrisyon at kung anong nagagawa ng ating genes sa paano na metabolize o paano natin pinoproseso ang ating mga kinakain. And at the same time, kung ano yung nagagawa ng ating mga kinakain sa ating genetic makeup. Ang nutritional genomics ay patuloy na pinag-aaralan at sinasaliksik, pinapaunlad ng DOST-FNRI, nakikipagtulungan sa marami po nating mga stakeholders at patuloy po na naghahanap ng aming mga magiging kaagapay para mapalago ang nutritional genomics research sa ating bansa. Para sa bayan, sabi nga nila, it's a science for the people.